All right, so in a previous video we went over kind of the nuts and bolts of DNA replication, the enzymes that are involved, and how that DNA is copied. This video, what we're going to discuss are some of the differences between prokaryotic and eukaryotic DNA replication, because there are some portions of DNA replication that are going to be a little bit different associated with bacteria versus eukaryotic cells. All right? And there's going to be some things that are the same, and we saw some of that in the previous video. So both prokaryotic and eukaryotic organisms are going to have semi-conservative replication. All right? What that semi-conservative replication means is you've got one strand that is the old strand. I'll put that in quotes. And that's going to serve as the template. So that's the strand of DNA that the enzymes use to determine which nucleotides should be inserted. Then there's going to be the new strand. All right? And that's what's copied or replicated in the process of DNA replication. So both prokaryotic and eukaryotic cells have semi-conservative DNA replication. So that's the same between them. You're also going to have bi-directional DNA replication. When I was talking about the replication bubbles that open up, you'll get replication bubbles in both prokaryotic and eukaryotic organisms. And what you're going to have some place where the DNA replication is going to start, and that's going to be the origin of replication. And then you're going to have DNA synthesized in both directions, okay, to make a complete copy. So you've got bidirectional replication in both prokaryotic and eukaryotic organisms, all right? And the other thing that's going to be the same, and this kind of relates to the bidirectionality a little bit, is you're going to have the leading and lagging strands that we went over. Remember, the leading strand is synthesized in one long piece. The lagging strand is where the Okazaki fragments come in. So you're going to have Okazaki fragments associated with both prokaryotic and eukaryotic organisms. All right? So you're like, okay, great, I watched all that, what's different? There are some things that are different, and with prokaryotic cells, there's going to be a single origin of replication, and that's often referred to as the ORI. So that ORI is just the origin of replication. And the reason there's only a single origin of replication associated with prokaryotic cells, most prokaryotic chromosomes, and it's double-stranded, so I'll draw another one in here, are going to be circular. So what happens with that bidirectional bubble, you've got a single origin of replication, and when that double helix comes apart, you'll get replication in both directions, and eventually that replication bubble will meet, and you'll get the separation of the two complete genomes associated with the prokaryotic. Okay? Eukaryotic DNA replication, you're going to have multiple origins of replication. And that's because with eukaryotic cells, the chromosomes are going to be linear. So in order for that DNA to be replicated in the eukaryotic cell and for the process to not take forever to happen, okay, you're going to have replication happen at multiple places, okay? So you've got multiple origins of replication. That's because the chromosomes are linear. The chromosomes with prokaryotic organisms are going to be circular, all right? And that circular chromosome generates another problem with prokaryotic organisms. Because you've got the circular chromosome, you've got issues that are induced with that structure when the DNA is being replicated. And what is introduced is known as supercoiling, all right? Now, the bacterial chromosome to start with, it already has some negative supercoiling associated with it. But when you open up that origin of replication and start that process of all those proteins coming in to make a copy, you're stretching that essentially closed circle, and it's going to put torsional strain on the DNA molecule further downstream from where the origin of replication is, okay? So that supercoiling, probably should have done this a little bit differently, but that's okay, all right? The supercoiling is going to induce torsional strain on the DNA molecule. So what happens is the bacterial cell has a way to deal with that. And how it deals with that is using topoisomerases, okay? The topoisomerases in prokaryotic organisms are going to work to relieve the strain that's associated with DNA replication, all right? And what I need to tell you, eukaryotic cells also have topoisomerases, okay? Yeah, great. You just told me these were bacterial. Now you're telling me it's eukaryotic. There are different types of topoisomerases, okay? In eukaryotic, it's type 1 topoisomerase. And the thing about type 1's topoisomerase, that's a mouthful, it doesn't require ATP to happen. Because you've got a linear chromosome, the torsional strain associated with opening that up, you're still going to have some strain on the DNA molecule, but it's not going to be at quite the level that you do when you've got a closed circle, okay? So the type 1 topoisomerase doesn't require ATP, and it relieves positive and negative supercoiling that happens, all right? So with prokaryotic organisms, our topoisomerase is going to be type 2 topoisomerase. And this one is going to require the addition of ATP. So it's going to be ATP dependent, okay? And this is also going to relieve positive and negative supercoiling. But the type 2 topoisomerase is going to be unique to bacteria. And interestingly, even though plants are eukaryotic, plants also have type 2 topoisomerase, okay? So that topoisomerase type 2 is going to be important in relieving the torsional strain that's associated with the DNA molecule with DNA replication, okay? So those are just a few of the differences associated with prokaryotic and eukaryotic replication. If you have any questions, please let me know.